Good morning, students. Let us start chapter number 2, Sexual Reproduction in Flowering Plants. Flowers do not exist for us only. They are the sexual reproductive parts of the plants. These adaptations are meant for the successful sexual reproduction. What we enjoy gazing myriads of flowers, we swoon over scents and perfumes, and the rich colors are all because of the adaptations for the sexual reproduction in a plant. These adaptations ensure the formation of the end products of the reproduction, that is, seed and fruit. Flower is a fascinating, astonishing organ of a plant. We all are attracted by the flowers, isn't we? We have a very close relationship with the flowers since time immemorial. We'll study morphology, structure, and process of sexual reproduction taking place in the flowering plants, that is angiosperms. Flowers are the objects of aesthetic, ornamental, social, religious, and cultural values. Flowers have always been used as symbols for conveying feelings like love, affection, happiness, grief, mourning, etc. Let us see what flowers are to a biologist. To a biologist, flowers are morphological and embryological marvels. Typical structure of flower consists of four valves. Two types of components are there: essential and non-essential. Essential includes stamen and pistil, which are directly involved in reproduction. Stamen are the male reproductive parts of the flower, while pistil is the female reproductive part of the flower. Non-essential parts include sepals and petals, which are not directly involved in the reproduction. Sepals, role of sepals is to protect the flower in its bud condition, while role of petals is to attract the pollinators so that pollination may occur. In both the cases, they are indirectly involved in reproduction. Hence, they are known as non-essential parts of the flower. Let us see pre-fertilization structures and events of the flower. Several hormone and structural changes are initiated that leads to the differentiation and the development of the floral primordium. Inflorescences are formed with the floral buds, which then flowers. In a flower, male and female reproductive structures develop. Male reproductive structure represents androecium, which is a whorl of stamen, while female reproductive structure represents gynoecium, which is a whorl of pistil. It's time to study stamen, that is androecium. It majorly consists of two parts: anther, a bag-like structure, and a filament, which is a long slender stalk. Anther is a terminal bilobed structure, while filament is a slender stalk with which the anther is attached to the floral parts. The proximal end of the filament is attached to the thalamus or petals, and the distal end with the anther. Number and length are variable when we talk about stamen. There are some flowers with only single stamen. There are few flowers with more than one stamen. Also, the length varies. Also, the shape of the anther varies in different species. A typical angiosperm anther is bilobed with two theca in each lobe, hence known as dithecous. A longitudinal groove runs lengthwise, separating the theca. It is made up of connective tissues. There are various types of tissues in the transverse section of the anther. Let us see them. The bilobed nature of the anther is very distinct in the transverse section. Let's locate the stamen in a flower. Stamen, the male part, located in the flower. You can see the red bag-like structure, that is anther, and a green slender filament. Now we know this filament is meant for what? It is meant for the attachment of this anther with the floral parts. It may be petal or it may be gynoecium. So the anther will produce pollen grains which will be the carrier of the male gametes which are necessary for sexual reproduction. Structure of anther, as we have seen, it is a four-sided structure. It consists of 4 microsporangia, 2 in each lobe. These microsporangia develops further into pollen sac. Let us look the structure of microsporangia. Each typical microsporangia appears nearly round in shape. It is surrounded by 4 wall layers. Outermost epidermis, inner to it endothelium, inner to it are the middle layers. It may be one or several. The innermost layer is tapetum, which forms the basic structure of the microsporangia. It nourishes the pollen grain. Cells of tapetum possess dense cytoplasm and generally have more than one nucleus. In young anther, group of compactly arranged homogeneous cells are known as sporogenous tissue. Each spore will undergo microsporogenesis and forms the microspores. Sporogenous tissue undergoes microsporogenesis. Let us have a look on this process. What does it mean? The synthesis or the formation of microspores is known as microsporogenesis. Sporogenous tissue, the cells are known as pollen mother cells, that is PMC. They undergo meiosis and forms 4 microspores. Each one is a potential pollen or a microspore mother cell that will gradually converts into the male gamete. 4 microspores are referred to as microspore tetrad. As the anther matures, it dehydrates and the tetrad dissociates into pollen grains. These pollen grains are then released outside from the dehiscence of the anther. Let us see how the microspores are synthesized. We can observe the stamen. This is the thin filament. This is anther. We can observe the 4 microsporangia. This is the innermost layer tapetum, which forms the microsporangia. When we see the cross section of the tapetum, there are so many sporocytes. Sporogenous tissue, what we talked about, they will differentiate and develop into pollen. First of all, they will differentiate into 4 microspores, hence known as microspore tetrad. As the anther matures, it dehydrates and the tetrad dissociates and all the pollen grains are separated. They are ready for the release from the anther. This pollen is a potential microspore mother cell which further divides to form male gametes. Anther dehisces by the breaking of endothelium as well as epidermis during the release of the pollen from it. This is the way how the pollen are released. Further topics such as pollen grain structure, their importance, allergies, viability of pollen, pistil, megasporangium ovule, Microsporogenesis, embryo sac in the upcoming lectures. I hope you are enjoying the lectures and able to learn the facts about the sexual reproduction in the flowering plants. So, till then, keep studying.